This is Holy Cross mock for component 1 and it's mock 1, question 4. Here's a set of diagrams that, if you've done enough questions, you will immediately recognise. We have got respiration in a yeast cell, and we've got aerobic respiration going down there and the 4 stages that you know, and we've got another little step, possible anaerobic in the absence of oxygen. Name the stages, pretty straightforward hopefully. A is our glycolysis, breaking our glucose down into pyruvate. C is a cycle involved in respiration, there's only one of those, that's going to be our Krebs cycle. We missed out B. D, that's our oxygen being attached to the hydrogen to make water. So that's electron transport chain, oxidative phosphorylation, that stage. So we have got electron transfer chain, or I'm going to put oxidative phosphorylation. Oxidative phosphorylation. Name the compounds. So P comes off turning a 3 carbon compound into a 2. So there's a clue. So it's carbon dioxide, and also carbon dioxide comes off when we are turning our 6 into a 4, so it's going to happen twice there. And also we know about anaerobic respiration, this being ethanol here, being a 2 carbon, so we've lost a carbon there as well. So that all ties in with it being carbon dioxide. And as it says name, I'm going to give it its name, but I'm sure you'd get the mark for CO2. We mentioned Q being ethanol, so anaerobic respiration in yeast, so not in humans, in yeast, producing ethanol. R, where's R? There we are. Final electron acceptor in respiration, it's going to be oxygen, being combined with hydrogens coming off and turned into water, which is S. So we've got oxygen and water. Name the carrier molecule. That takes hydrogen from these various stages to stage D. Here you probably can get away with the initials because it's so common. It's nicotinamide adenine dinucleotide, but actually today I'm just going to put NAD. You could put NADH or reduced NAD and so on, but you say it delivers hydrogen, so the actual base molecule is NAD. Over the page. Get rid of that. During stage D, an electrochemical gradient is created across the inner mitochondrial membrane. So just a reminder, there's a mitochondria, that's the outer membrane, inner membrane, intermembrane space, and you might have these projections here, crista or cristae if there's a plural. And we have got matrix inside. How the components of the membrane use the hydrogen ions delivered by the carrier molecule to create the electrochemical gradient. Well, the components of the membrane, so things inside that membrane. And when you zoom into that membrane, the components that you expect to see will be kind of electron carriers and pumps and things like that. So this is a story of chemiosmosis, and that's what we're hoping to get from you. So this is just going to be energy from the electrons to pump protons. Energy from the electrons pump your protons, or H plus or hydrogen ions, across the membrane into the intermembrane space. So you have got hydrogen ions being pumped across there using the energy from the electrons. Using letters from the diagram, complete the following to show a process that takes place in the matrix that maintains this electrochemical gradient. Well, with not many letters to go at, the answer is R going to S, and underneath is the explanation. So, why the process you described affects the electrochemical gradient across the membrane. So, these hydrogen ions are going to drop back down, and the last part is explaining how ATP is generated in that step. But if we remove these hydrogen ions and put them into water using oxygen, then that will maintain the gradient. So if we are pumping and making lots of hydrogen ions there, these are dropping back through, but as you can imagine, as more drop through, the gradient becomes less. So if we get rid of these by making them into water, then that will maintain the gradient. So it removes protons. So it removes these hydrogen ions, protons, and so this gradient is maintained. Explain how this gradient subsequently leads to the formation of ATP. So when these drop back through this membrane, they drop back through a protein, which is also an enzyme, and you'll know the name of this. So protons flow down this gradient or diffuse back, so protons diffuse down a gradient. We've made sure that we know it's passive and diffusing a gradient through ATP synthetase, and in that, what's that going to do? That is going to to make ATP from ADP and inorganic phosphate. So most people got above 10 in that question, so it was a well answered question, and if they did lose marks, it was generally in this part of it.